Welcome back to this channel called Learn Bio with Janet. Today we'll be discussing the formation of urine in the kidney. It is interesting to find out how the kidney manages to separate the toxic waste substances from the useful components of blood. This lesson involves the KBSM biology syllabus for Form 5. Chapter 3 on coordination and response, and the subtopic is 3.4. Now, in the last lesson, we have already discussed the parts of the nephron, which is firstly the Bowman's capsule that's shaped like a cup at the beginning of the nephron, and then secondly the proximal convoluted tubule, which is the second part of the nephron, and it is a coil, the coil part of the nephron. The third part is shaped like a U shape called the loop of Henle, and the fourth part is the distal convoluted tubule. That's again a coiled portion of the nephron. Okay, and then this nephron is connected to a collecting duct that collects the urine formed at the end here. Then for the blood vessels, we have the afferent arteriole that brings the blood or transports the blood into the glomerulus. And glomerulus is a cluster of blood capillaries or a group of blood capillaries that is located inside the cavity of the Bowman's capsule. Then the blood flows out through the efferent arteriole, and then efferent arteriole will branch out to form the capillaries that surround the nephron. Okay, the network of blood capillaries that surround the nephron. Then after that, the blood will flow back to the heart through the venule and the veins. So this is the system of blood capillaries that surrounds the nephron. To start off this lesson, let's just look at the overview of the processes that cause the urine to be formed. So there are three processes that we must know, and that is ultrafiltration, reabsorption, and secretion. Okay, ultrafiltration, reabsorption, and secretion that occurs in the nephron from the Bowman's capsule to the end of the nephron. So the different processes occur in different parts of the nephron. Now the first process is ultrafiltration, which we will study in more detail in the next slide. So ultrafiltration occurs at the glomerulus when substances in the blood are forced out into the Bowman's capsule. That means substances from the blood will come out and move into the Bowman's capsule. Then reabsorption occurs in the proximal convoluted tubule here, proximal convoluted tubule, loop of Henle here, loop of Henle here, and the distal convoluted tubule here, distal convoluted tubule, and also in the collecting duct that collects the urine here. Okay, so there are four places where reabsorption occurs: proximal convoluted tubule, loop of Henle, descending limb here, and ascending limb, and then distal convoluted tubule and the collecting duct. So for reabsorption, substances that are useful to the body are reabsorbed back into the blood. So it goes back into the blood. For example, all glucose is reabsorbed by active transport back into the blood from the nephron, or we can call this the renal tubule. Nephron or renal tubule. Renal means something to do with the kidney, so nephron or renal tubule. So the renal tubule includes the proximal convoluted tubule, the loop of Henle, distal convoluted tubule, these three parts. Okay, so we will discuss reabsorption also later. Now the third process that leads to the formation of urine is secretion. Secretion is the movement of the waste products like urea back into from the blood into the nephron. Okay, sorry, from the blood into the nephron. So the direction of movement of substances is same like ultrafiltration, from blood into nephron. Secretion is also from movement of the substances from blood into nephron, the unwanted substances that have not yet been excreted. Some of them may still be in the blood, so they are secreted in the distal convoluted tubule especially. Okay, so we'll be talking about each process one by one. So let's start with ultrafiltration. Let's be able to discuss how ultrafiltration occurs. So here are the notes for the introduction. What are the three processes that lead to the formation of urine? Ultrafiltration at glomerulus, in the cavity of Bowman's capsule, and constituents of blood are filtered out under high hydrostatic pressure from glomerulus out of blood into Bowman's capsule, which is part of the nephron. B, reabsorption occurs along the renal tubule, meaning the other parts of the nephron, okay, below the Bowman's capsule. So useful substances must be reabsorbed from the nephron back into the blood at the proximal convoluted tubule, or PCT, loop of Henle, distal convoluted tubule, and collecting duct. And C, the secretion at distal convoluted tubule. Okay, so secretion is the third process. Waste products are pumped out from the blood into the nephron, into the distal convoluted tubule. Now the first process that's involved in the formation of urine is called ultrafiltration. Ultrafiltration. It occurs at the glomerulus. Now, what is the adaptation of the blood vessels for ultrafiltration to occur? So looking at this picture, now this center part here is the glomerulus, which is a cluster of blood capillaries or a mass of blood capillaries in the cavity above the Bowman's capsule. Okay, so what is the adaptation of the blood vessel? Now, if you look at this blood vessel here, this blood vessel is called the afferent arteriole. The afferent arteriole has is wider or has a bigger diameter. Or we say it is bigger. It has a bigger diameter or is wider than the efferent arteriole. So afferent arteriole brings or carries the blood into the glomerulus. Efferent arteriole carries the blood out of the glomerulus. Now, the afferent arteriole is wider, so it brings more blood, it carries more blood into the glomerulus. But less blood flows out through the efferent arteriole, which is narrower. Therefore, the blood will accumulate in the glomerulus. And as it accumulates, it will cause the pressure inside to increase. We call this the hydrostatic pressure. So, in the glomerulus, the blood is at high hydrostatic pressure. Remember this word, this is a key word. High hydrostatic pressure. Now, when you write your answer, you must use these words. High hydrostatic pressure. Don't say high blood pressure. High hydrostatic pressure. So, when the blood is at high hydrostatic pressure, it will be forced out of the walls of the capillary, because glomerulus is actually blood capillary. It's made out of blood capillaries. So the blood is forced out through the wall of the capillaries into the Bowman's capsule. Now this process whereby the blood is forced out is called ultrafiltration, because many components of blood are filtered out. What do you mean by filtered out? It means that the soluble components and the fluid will be forced out, but it will leave some components of blood inside the blood capillaries. The bigger components that cannot pass through the wall of the blood capillaries will stay inside the blood. That's called filtration. It's a separation of some of the components of the blood. Some components, like red blood cells, will stay in the blood capillary because it's too large to come out. Okay, other substances or other molecules or cells that are too big, the cell that is bigger and can't come out are the platelets also. It's a fragments of cells. Platelets are fragments of cells. And also molecules like the plasma proteins that are bigger in size. They cannot pass through the pores of the wall of blood capillaries because the pores are too small for them to pass through. So when ultrafiltration occurs, many components of blood are filtered out. Let's look at the components that are filtered out. Now, the fluid that is formed in the Bowman's capsule is called glomerular filtrate. Okay, say this word, glomerular filtrate. Glomerular filtrate is not in the glomerulus. Please take note. It has come out from the glomerulus it actually comes out from the blood. So these are the components of blood that are filtered out: water, urea, glucose, amino acids, salts, sodium ions, chloride ions. Notice that all these components are made up of smaller molecules, so they can pass through the walls of the blood capillary here, through the pores in the walls of blood capillaries. But this glomerular filtrate will not contain plasma proteins, platelets, or erythrocytes, because these are too big to pass through the wall of the blood capillaries. So they remain in the blood. Plasma proteins like globulin, albumin, fibrinogen will stay inside the blood. Platelets and erythrocytes too. And this is the meaning of filtration, a process that separates solid matter from a liquid. For example, it separates the erythrocytes and the platelets from the plasma, some parts of the plasma like water, urea, and all that, by causing the liquid to pass through the pores of a filter. The filter in this case is the capillary wall. So now we understand the meaning of the word ultrafiltration. Ultra here means in a very extreme form, because this filtration occurs on a microscopic level. It's at a very small level. So it's called ultrafiltration. Here are the notes about what I discussed just now. So I put in the notes to help you be able to write it, especially for essay questions and structured questions. Okay, you have to know how to explain it. So this is the way we explain ultrafiltration as what I discussed just now. I will go through this very quickly. Ultrafiltration occurs at the glomerulus, in the cavity of the Bowman's capsule, and the afferent arteriole has a larger diameter than the efferent arteriole. So this is an adaptation of the arterioles. Thus, the blood in the glomerulus is under high hydrostatic pressure, and the high hydrostatic pressure in the glomerulus forces out some fluid. It's something like plasma, blood plasma, through the wall of the blood capillary, or through the walls of the blood capillaries, into the Bowman's capsule. This process is called ultrafiltration, and in this way it forms the glomerular filtrate in the Bowman's capsule. Composition of glomerular filtrate: only the smaller molecules that can pass through the blood capillary wall will form the glomerular filtrate. So these substances are water, glucose, amino acids, salts like sodium ion and chloride ion, and urea. Let us look at a common question asked in this section of ultrafiltration. State 3 components of blood that are not found in the glomerular filtrate. So what are the 3 components as we discussed just now? It's the substances or the molecules that are bigger, right? For example, erythrocytes, red blood cells, are big in size, so they cannot pass through the walls of the blood capillaries. Platelets, or fragments of cells that are involved in blood clotting, and plasma proteins such as fibrinogen and albumin. The plasma protein molecules are too big to pass through the walls of the blood capillaries. So explain your answer in question 1. As we said just now, these components of blood are too large to pass through the pores, or just to pass through the blood capillary wall. Let's refer back to the same diagram of the nephron. We have already discussed the first process that causes the formation of urine, and that process is ultrafiltration. So ultrafiltration occurs when certain components of the blood are forced from the blood, they are forced into the Bowman's capsule. So this forms the glomerular filtrate, which contains water, salts, glucose, amino acids, and urea. And we can see here that there are some useful components like glucose and amino acids. So the body will get back some of these useful substances through reabsorption. So reabsorption is the process whereby certain components in the glomerular filtrate are absorbed back into the blood. They move back into the blood from the renal tubule. Renal tubule means the nephron, the the part of the nephron, no, below the Bowman's capsule here. So in the, now what is this area called? Proximal convoluted tubule. So in the proximal convoluted tubule, all glucose, all amino acids, vitamins are reabsorbed into the blood through active transport. That means it needs energy for this reabsorption to occur. The reabsorption of glucose, all, remember, all the glucose need to go back into the blood at this proximal convoluted tubule. So no glucose will be found in the urine formed at the end here of the nephron, because all the glucose has been reabsorbed back into blood at the proximal convoluted tubule here. Okay, water will be reabsorbed by osmosis. Sodium ions reabsorbed by active transport. So most of the substances are reabsorbed by active transport. Chloride ions are different. They are reabsorbed by passive transport or diffusion back into blood. But most of the substances are reabsorbed by active transport. So when these substances are reabsorbed, it will cause the solute concentration, that means the concentration of dissolved substances in the blood to increase, and this will cause water to diffuse into the blood capillaries naturally by osmosis, which does not use energy. The water diffuses from region of low concentration of solutes in the nephron back into the blood, which has high concentration of solutes like glucose, amino acids, and the salts that have been actively transported back. Okay, let's go on to the second part. Now the second part is the descending limb, the descending limb of the loop of Henle here. It's a U-shaped loop. Okay, so there's reabsorption of water by osmosis at the descending limb as the fluid, the glomerular filtrate, flows down here. How about the ascending limb? Now in the ascending limb, there's reabsorption of salts using energy. That means by active transport. Okay, salts are reabsorbed by active transport. Water is always reabsorbed by osmosis. Right, and then what about the distal convoluted tubule? So reabsorption at the distal convoluted tubule and the collecting duct for salts and water depends on what the body needs. If the body is lacking salt, then salt has to be reabsorbed. If the body is lacking water, water will be reabsorbed back into the blood. It will not be excreted. So if water is reabsorbed back, that means the urine will become more concentrated and have less water in it. So these processes, the reabsorption of salts and water at the distal convoluted tubule and the collecting duct, are controlled by the hormones aldosterone and ADH. ADH is antidiuretic hormone. We'll be discussing this in the third video, how these hormones help to control the reabsorption of salts and water. Okay, now, so we're finished discussing reabsorption, and we might as well discuss secretion also. Okay, never mind. I'll leave it to the next slide. Next few slides now to discuss secretion. So let's read the notes on reabsorption first. Here are the notes on reabsorption. Just let me go through very quickly so that we have a clear understanding of this process. Reabsorption in the renal tubule or the nephron. Reabsorption of useful substances occurs from glomerular filtrates in the nephron back into the blood. They came out of the blood, now they're going back into the blood. All along the renal tubule. Okay, these are the locations where reabsorption occurs. BCT means proximal convoluted tubule here. Low is the loop of Henle. DCT and CD is the collecting duct. DCT is distal convoluted tubule. Okay, dissolved substances move across the walls of the renal tubule back into the blood capillaries. By the way, all this short form you have to spell in full whenever you answer the question in the exam. Now, in the proximal convoluted tubule, that's where most of the reabsorption occurs, the reabsorption of useful substances. All, remember this word is all, I must remember this. All the glucose and amino acids are reabsorbed by active transport. Sodium ions also are actively pumped into the blood capillaries, again by active transport. But chloride ions are reabsorbed passively by diffusion. Water is also by passive processes, water diffuses into the blood capillaries by osmosis. It's also a passive form of transport. When the concentration of solutes in the blood capillaries become higher, because you have amino acids and glucose and salts, no, as the solutes in the blood capillaries. So this will cause the concentration of solutes in blood capillaries to become higher than that of the glomerular filtrate. So water will diffuse from the glomerular filtrate into the blood capillaries. Okay, some further notes on reabsorption. In the loop of Henle, water is reabsorbed by osmosis. Salts like sodium and chloride ions are reabsorbed by active transport. Here, in the descending limb, water is reabsorbed. In the ascending limb, salts are reabsorbed. Okay, and then in the distal convoluted tubules and collecting ducts, so here and here, water and salts are reabsorbed, but the amount of water and salts reabsorbed depends on what the body needs. So if body lacks water, antidiuretic hormone is secreted so that more water is reabsorbed from the urine that's formed here and here back into the blood. If the body lacks salts, person didn't eat enough salts, aldosterone is secreted from the adrenal gland so that more salts are reabsorbed into the blood. We'll be discussing this in the third video. Here is a table of the summary of reabsorption. So I'll not go through this again because we have discussed it already, but you can copy down this table if you feel that it helps you. Some students learn better when the information is tabulated. So you can copy this, screenshot it and copy it, or pause the video. We have already discussed two processes involved in urine formation. Firstly, there's ultrafiltration at the glomerulus and the Bowman's capsule. Second process is reabsorption at the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct. Now the last process, the third process involved in the formation of urine, other than ultrafiltration and reabsorption, is secretion. Let's say this word, secretion. Secretion is the movement of the waste products such as urea, uric acid, ammonia, excess hydrogen ions, excess potassium ions from the blood into the nephron, and this process occurs mainly in the distal convoluted tubule, the last part of the nephron. So these substances were still in the blood and were not filtered out of the blood at the first phase, in the first phase of ultrafiltration. So they were still in the blood capillaries. So this is another chance to get them out, and that is by secretion, which can occur by active transport or by diffusion, which is a type of passive transport. Okay, so for urea and uric acid, they are secreted by active transport. But for drugs and toxins, is by diffusion without the use of energy. Okay, so that's why if a person takes drugs, the drugs will be found in his urine when they do a urine test, and that will be proof, positive proof that the person took the drugs. Okay, so that's used in the test to test whether a person has taken drugs or not. So secretion is to get rid of the waste products more quickly by using energy to move them. Like urea is moved across from the blood into the nephron in the distal convoluted tubule mainly. Let's discuss a HOTS question on reabsorption. So you are given a diagram here, all right, that shows the nephron, and this part is circled to show you the wall of the nephron and the blood capillary beside it. All right, so based on the diagram of cell X in the wall of the renal tubule or nephron, state 3 adaptations of cell X and the renal tubule for active transport of glucose. So based on this diagram, what do we see here? That we can that is the adaptation, or that are the adaptations of cell X. Okay, for example, can you see these projections here? It's something like the projections on the epithelial cells of villus. So the epithelial cells of the villus in the small intestine has a lot of this projection too. What do you call them? Microvillus. So what is microvillus used for? So you apply what you have studied in chapter 6 about absorption in the villus. Okay, so the microvillus here increases the what? Yes, the total surface area of the cell for faster absorption of glucose into it. Okay, so the first answer, cell X has many microvilli, all these tiny projections, to increase the total surface area for reabsorption of glucose, okay, into the cell. Now the next point are the mitochondria. Okay, now you have to recognize that these organelles are mitochondria. What are they used for? Generate energy through cellular respiration. Okay, for what purpose? For active transport, correct, of glucose. Cell X has many mitochondria to generate ATP, which provides energy for active transport of glucose. Now the third point is that the wall of the nephron, how thick it is? Is it 1 cell thick, right? So the wall of the tubule is very thin. The wall of the tubule is very thin, only 1 cell thick, for faster transport of glucose across it. So glucose just needs to go past this wall, okay, into the blood. Just 1 layer of cells. Okay, so these are the 3 adaptations: microvilli, mitochondria, and thin wall for the proximal convoluted tubule. Here are the notes on secretion in the renal tubule. Secretion is the process of removing waste substances from blood into the renal tubules or nephrons. So actually, some waste substances have filtered out during ultrafiltration into the Bowman's capsule. But if the waste substances did not filter out here, they can still be secreted from the blood into the renal tubules at the distal convoluted tubule. Okay, so this process is the opposite of reabsorption, because the substances are moved from the blood into the nephron, whereas in reabsorption, the substances are moved from the nephron into the blood. Secretion occurs in the distal convoluted tubule here, collecting duct also, by passive diffusion and active transport. Okay, so what are the substances that are secreted into the renal tubule? Excess hydrogen ions that are not wanted, excess potassium ions, excess ammonium ions, urea, creatinine, uric acid also, ammonia, toxins and drugs. All these are unwanted, need to be removed. Number 5, purpose of secretion is to remove the toxic wastes from body more quickly, to regulate the levels of ions such as hydrogen. So if there's too many hydrogen ions in the blood, then the kidney will excrete these hydrogen ions, secrete them into the distal convoluted tubules, so that it can manage or regulate the blood pH. Because if the more hydrogen ions the blood has, the lower the pH. So if it's too low, then the kidney will excrete it, so that the blood pH can increase back to normal. Finally, after the 3 processes have occurred, that is ultrafiltration at the glomerulus, reabsorption all along the renal tubule, and also secretion mainly at the distal convoluted tubule. So after these 3 processes have more or less finished at the nephron, the urine will be formed here. Okay, so the urine is formed at the junction with the collecting duct, which helps to collect urine to the pelvis. So by the time the filtrate or renal fluid reaches the collecting duct here, 99% of the water has been reabsorbed back into the blood. Only 1% of salt and 1% of water and a little salt is left and forms the urine here, which will flow down the collecting duct here to the pelvis and then to the tube, tiny tube called ureter, and then to the bladder for storage. When it is full, then the bladder will expel the urine or excrete the urine through the urethra. Okay, so urine is a fluid that contains 96% water, 2.5% nitrogenous waste products, such as urea, uric acid, ammonium, and creatinine, and also mineral salts. Lastly, let's discuss a structured question on the formation of urine. Question 4. The table below shows the approximate composition of 3 different fluids in different parts of the kidney. So the components that are analyzed are water, glucose, urea, plasma proteins, and amino acids. And the 3 fluids where these components are found, and the 3 fluids that are analyzed are blood in the glomerulus, inside the glomerulus here. It has not filtered out yet. This blood is still inside the blood capillaries. Then the glomerular filtrate in the Bowman's capsule here, inside the Bowman's capsule. So this is the filtrate that is formed after ultrafiltration. And then lastly, urine, the collecting duct here. Okay, now state the process by which the glomerular filtrate is formed. Yes, it's ultrafiltration. Explain why no glucose or amino acid is present in the urine, although they were present in the glomerular filtrate. Now you can see here that there's some glucose in the blood and in the glomerular filtrate, but none in the urine. And same for amino acids. Present, present, not present. So something happened from this point to this point along the way in the nephron. And what is that process that occurred to the glucose and the amino acids? Yes, it's reabsorption. Okay, so the glucose and amino acids were reabsorbed back into the blood right now from the glomerular filtrate in the proximal convoluted tubule. Let's read. All the glucose and amino acids have been reabsorbed. You can write by reabsorption. The process is important. Back into the blood in the proximal convoluted tubule by active transport. Okay, 2 marks. So all the glucose and amino acids have been reabsorbed back into the blood. 1 mark. In the proximal convoluted tubule, maybe 1 mark, and active transport 1 mark. Going on to question C. Explain why plasma proteins are not present in the glomerular filtrate. So plasma proteins did not filter out from the blood and did not go into the Bowman's capsule. Okay, so it's not, the plasma proteins are not present in the glomerular filtrate in the Bowman's capsule. So the reason is because they are too large, okay, too large in size to pass through the tiny pores of the blood capillary wall. They couldn't go through the blood capillary wall, which was smaller. The pores were smaller in size. Okay, D. A patient's urine contains glucose. What disease is he suffering from? Explain the reason for this symptom. So once the person's urine has glucose, it's not normal, because normal people don't have glucose in their urine. So as the as it is stated in Malay, penyakit what? Kencing manis. That means the urine contains glucose. Please don't write the urine tastes sweet if you're asked to give a symptom of this disease. What disease is it? Diabetes mellitus. Or in Malay is penyakit kencing manis. So if you're asked to give a symptom for diabetes mellitus, I have some students writing the urine tastes sweet. So you cannot write taste sweet because nobody is going to taste the urine at all. So the urine contains glucose is one of the symptoms of the disease. Now, his pancreas did not produce enough insulin to convert all the excess blood glucose to glycogen. So his blood sugar level is high. That's the reason for the glucose in the urine. When his blood sugar level is higher than normal, the kidneys are unable to reabsorb all the glucose back into the blood in the proximal convoluted tubule. So some of the glucose will be excreted in the urine due to the high level of glucose in the blood. So not all will be reabsorbed back into the blood in the proximal convoluted tubule. Okay, so that ends our session today for this lesson. Thank you for watching. Please share, like, and subscribe. And in the next video, we are going to discuss the role of hormones in osmoregulation. Goodbye for now.